It's nice to be here today. The area of molecular oncology and molecular pathology is moving forward at an incredibly rapid rate. We now have a better understanding of human disease, and we're building a battery of targeted therapies to improve the health outcomes of our patients. And in order to do that, we've built some fantastic technologies. So now we have the challenge of which question to ask when and how to ask it, which technology to use to ask that question. And we also have other challenges. In the United States, we call it reimbursement. Here in India, we call it out of pocket. So we need to make sure that our patients can afford to pay for the questions that we're going to ask, and that we're applying the right technology to answer those questions at the lowest cost. So today I'm going to talk to you about the MassARRAY system from Agena Bioscience. And as was briefly mentioned before, in May of last year, Sequenom split its two divisions, and the bioscience, or the technology division of Sequenom, became Agena Bioscience. So you can call us Sequenom, call us Agena. Charles and I have been with both companies for a long time. We answer to both names. All right, so Agena's only been around for 14 months, but of course the MassARRAY technology has been around for near on 20 years. So therefore, we have the energy of a brand new company, just like MedGenome has the energy of a brand new company. But we have the history and knowledge of a 20-year-old company. So we feel like we're in the best of two worlds, because we have a broad install base of instruments across the world, more than 300 instruments across the world. 30 to 40% of our instruments are used in clear style, LDT, laboratories, whatever you want to call it, depending on your regulatory nomenclature. Further, around about a third of our instruments are in commercial livestock. In fact, millions of livestock know who their daddies are because of us. They know who to give a kiss to on Father's Day. And the remainder of our instruments are doing more pure academic research. Now, the core workhorse of the MassARRAY system is MALDI-TOF mass spectrometry. We use mass spectrometry as a detector for nucleic acids. Now, mass spec is well recognized in the proteomic space for being incredibly sensitive, accurate, precise, and we apply those advantages to nucleic acid analysis. So using mass spectrometry as a detector of nucleic acids provides significant advantages. It's high throughput. These things fly incredibly quickly through mass spectrometry, and thus we can do up to thousands of samples per day. Unlabeled oligonucleotides, where we're detecting the nucleic acid itself, not detecting a fluorophore that's attached to the nucleic acid and making an assumption. Not having to subtract a fluorescent baseline. It means that we have incredibly swift panel design. And I was at a customer site yesterday, and they said, how swift? And we got pulled out 20 somatic mutations, put it through the assay design suite, and before we'd finished the next part of our conversation, about 8 minutes later, a design was sitting on my desktop, right there. So it is incredibly swift panel design. Give me sequence now, I'll have a design for you in half an hour, we'll have oligos on your desk tomorrow. So this works incredibly well in the space of personalized medicine. Designing content for each individual patient, right? So truly personalized medicine. For every patient, a unique panel being designed to track their disease. Highly robust on multiple sample types. We use everything from hair follicles out of cattle all the way through FFPE tissue, so on and so forth. Short amplicon design means we're reliable on clinical samples. And let's face it, clinical samples are degraded. Accept it. Don't fight it. FFPE is here to stay. So make sure your panel works on it. Highly cost effective, broad range of applications, and today I'm going to focus on the areas with most clinical relevance, being somatic mutation analysis in oncology, gene fusion detection. We're going to, in fact, I'll pass over to Dr. Cantor for ultrasensitive detection and liquid biopsy. and we'll also cover normal inherited diseases, so germline genotyping. But we do have a variety of other applications that can be performed on the instrument as well. Personally, I think this is going to be the next biomarker that truly moves into the medical space. But we won't discuss that today. We'll discuss what's already in the clinical space already. So a complete offering of products and services. We supply you with the technology. So that is the mass spectrometer and the upfront robotics to miniaturize your reactions and transfer them into the mass spectrometer. We provide you with a variety of applications, biochemistry and software packages, simple PCR-based biochemistry and software packages that are almost completely automated for analysis. And then we have our reagents, a variety of different panels, and I'm going to show you all different styles of panels, but because I work here in Asia, you might have noticed my Australian accent by now. Don't ask me about the cricket, I have no idea, right? But I work in Asia, I come to India regularly, we build content that is relevant for Asia, not just relevant for the United States. All right, so we have a variety of different panels, but we also have our general purpose reagents, such that you can design your own panel, your own content. You're in control of your destiny, right? And you run it on these general purpose reagents. So the entire workflow consists of basic miniaturized PCR reactions, followed by post-PCR processing, whether that be single base extension or a resequencing chemistry that we call mass cleave. You then transfer, see, biochemistry is in a liquid, and inside the mass spectrometer is a vacuum. We can't put a liquid into a vacuum. So what we do is we transfer the biochemistry onto a SpectroCHIP that I'll show you shortly, crystallize the nucleic acids such that they're in the solid phase, so that we can put them inside the mass spectrometer to acquire and detect the nucleic acid fragments in that reaction, and then data analysis. So time to data, from DNA to data, is a standard working day, and in fact, we're improving on that rapidly, in fact, and we have a number of panels that work on what we call our short protocol, which is a 5.5 hour DNA to data. And we'll be continuing to improve that because we recognize that turnaround time is critical for the clinical laboratory. The general process of the biochemistry that I'm going to tell you about today, which is our iPlex single base extension biochemistry, is that we start with short amplicon PCR. Ideally, we keep this 60 to 100 base pair PCR amplicon size such that it's highly robust. We can use longer amplicons if it's required for specificity, but generally we keep it in the short amplicon. Amplifying the region of interest. We're not actually detecting the PCR product with the mass spectrometer, so we've got flexibility in where we can place our PCR primers, right? Such that we have uniform PCR amplification conditions for all panels and all reactions within all assays within every panel. So all of our panels go on the same plate. They also go on the same chip. So you have this random access flexibility. Following PCR amplification, we anneal a primer adjacent to our target polymorphic or mutated nucleotide. We know the sequence of that primer because we manufactured it, or in fact somebody manufactured it. Oligos are a commodity. You can use any oligo provider, right? You don't have to use a GENA for your supply of oligonucleotides. But we know what its sequence is. Let's say, for example, it has 5 A's, 5 C's, 5 G's, 5 T's. We know the masses of each of those bases. So we know what the mass of that oligonucleotide is. And for simplicity, let's call it 5,000 Daltons. All right? And then what we do is we do a single base extension with a mixture of all 4 terminator bases. So in this particular instance where we've got both the C and the G alleles in this particular sample, we're looking for the mass of the primer, 5,000 Daltons plus the mass of a G, the primer plus the mass of a C. So we're looking for 5,329 Daltons or 5,289 Daltons. And they resolve as separate peaks in the mass spectrometer because they have a different mass. Mass spectrometry is measuring the molecular weight. It's not measuring the number of nucleotides, it's measuring the molecular weight of those nucleotides. There is no colour that is put there for your visual appeal. by a marketing department. Mass spectrometry is black and white. All right? So we see two peaks, one for the G, one for the C. If this is a normal germline heterozygote, those peaks will be 50-50 or 1:1. And we measure quantitatively those peak areas by measuring the area under the allele frequencies by measuring the area under the peak to a standard deviation of 1%. Now, not all clinical samples are pure, and this is an example of a somatic mutation, right? Where we have unbalanced, or it could be a copy number variation, right? Where we have unbalanced allelic frequencies, we have a higher proportion of the G versus the C peak. Of course, we can measure that. MALDI-TOF mass spectrometry. Matrix-assisted laser desorption ionization time-of-flight mass spectrometry. Whole stack of words, but let's work through it one by one so that we understand what's going on inside the mass spectrometer. Matrix-assisted. So we take a very small amount of our biochemistry, 10 nanoliters, in fact, of our biochemistry, and we deposit it onto our SpectroCHIP. On our SpectroCHIP, we have crystals of our matrix, right? So what happens is the matrix dissolves into that 10 nanoliters and crystallizes back out over the course of a couple of seconds. So we now have our matrix, our nucleic acids surrounded by our matrix, right? So that's the matrix assisted, because when we pulse the laser, we apply energy to that, and the matrix donates a proton to the nucleic acid, right? So it donates a positive charge and 1 Dalton. But it donates a positive charge and also moves into the vapor or the gaseous phase. So instead of being a solid crystal, our nucleic acid is now in the gaseous phase, such that using that charge, we can accelerate and measure the time of flight for it to get to the detector. That was actually pretty slow, because the time of flight is 3 nanoseconds. Slow it down a little bit in the cartoon, right? So it takes 3 nanoseconds to measure the time of flight of those molecules to reach the detector. And of course, the time of flight, because the version that we use, we're generally looking at what single charge species. The time of flight is directly contributable to the mass. Now, if it's 3 nanoseconds for one measurement, and it only takes a little bit of electricity, there's no polymer, it's a vacuum, so it's just a little bit of electricity, why not take more than one measurement? And that's what we do. We take 200 measurements of every reaction, measuring an experiment over and over and over again. What's the result of that? Quantitative accuracy, as well as sensitivity. So that's what mass spectrometry is giving us. Now, so far, it looks like we're only doing this one by one. And that's because it makes it simple for me to explain what's going on. But in fact, we do this in multiplex format. And this is an example of a 52-plex panel. I'm going to show you this panel later. So a 52-plex panel where all the alleles are potentially biallelic. So we've got 52 primer masses and 104 product masses that we're looking for across our mass spectrum. So that's the technology. Enough about technology. That's probably all I know about mass spectrometry. Maybe Charles can tell us a little bit more. Let's talk about disease states. Thalassemia. Don't need to tell anyone in India what thalassemia is, I'm sure. Severe disease, highly prevalent in this particular region of the world. But the mutations that occur in India are different to the mutations that occur in Malaysia, for example, and different to the mutations that occur elsewhere. So we're able to easily design panels with mutations that are relevant for your area, minimizing the cost, right? So this particular panel, 28 mutations. Malaysia's quite an admixture, in fact. It's a very mixture of different races in Malaysia. We've got our 14 mutation panel here in India, which is run by our partners at ILS, but also available to all of our customers. We have this 15 mutation well panel, which was designed for South Australia. And let me just focus on this one, because this one we did a significant orthogonal validation study on. So we took 231 samples, 115 of those were positive. You can see the spectrum of mutations if you're interested. What I'm interested in is that all 100% of the 115 samples were orthogonally validated with Sanger sequencing, right? It took a day's work to run the samples on the MassARRAY system. I can tell you it took a lot more for the validation study. we always do like to have that orthogonal validation data. Another challenge in this area of the world is congenital hearing loss. Congenital hearing loss affects 360 million people worldwide. 32 million of those are children, with a much higher prevalence in Asia. In China, there's 27 million people affected, and at the moment, newborn screening programs are screening around about 20 million newborns per year, and they're detecting more than 30,000 children. Now, these are genetic tests. There are about 5 genes involved in congenital hearing loss and specific mutations in those genes. So what does it mean? Well, it means that these children who get early newborn screening detection at the genetic level get early intervention. It's too late when the parents realise, maybe 18 months, 2 years old, 2.5 years old, that their child is struggling to hear them. At that point, their child is significantly developmentally delayed. And we have interventions available, whether it be linguistics interventions, whether it be sign language interventions, whether it be cochlear implant interventions. So these children are now getting treated much earlier and offsetting that delay that otherwise would have happened. Again, variety of different panels that we've developed. For BGI are running a 20 mutation panel in 4 of the genes. DAN are running 20 mutations in 5 genes. And our IFP are running 34 mutations in 3 genes, so on and so forth. But again, testament to the flexibility. Unfortunately, clinical groups don't seem to have consensus on what they want to run, right? So therefore they have a choice. They either just do what the technology company tells them to do, and I can tell you technology companies don't make the greatest clinical choices, or you use something like the MassARRAY, where we can design exactly what you want or what your clinicians want to run. Now we have users across the world. Quest Diagnostics and their lab at UMass, Edgins and Marzina have developed more than 35 LDTs on a single MassARRAY platform. Variety of different disorders, blood and immune disorders, Ashkenazi Jewish panels, pharmacogenomics panels, so on and so forth. And what do they like about the MassARRAY system? They can run all of these panels at the same time on the same plate, on the same system. So today they get 3 samples for panel 1, nothing for panel 2, 5 samples for panel 3, and all just goes onto the same plate, and off we go. They don't have to run everything individually. And when you have a large number of rare disease LDTs, that's what you want. Otherwise, you're either wasting labour to run just 1 or 2 samples every day, or you're batching and you're wasting time to get the result back to the patient. So what gives us that random access flexibility? So these are our SpectroCHIPs. We have 3 formats. We have a 24 pad, a 96 pad, and a 384 pad. They look like a microarray. They're nothing like a microarray. They're not specific to your experiment. They've got no oligonucleotides on them. It's a solid surface, pre-spotted with that proton donating matrix that we apply the biochemistry to. The test itself, the specificity of the test, is all in your liquid biochemistry. So that means that on the one SpectroCHIP, you can add 1 sample for a 1-well panel that might be looking at a somatic mutation. You can add another sample for an 8-well panel that might be looking at a series of rare germline genotypes, cystic fibrosis or something, for example. You can have 3 samples on another panel that has 2 wells until you fill up the chip, and you don't have to fill up the chip today. You can use half of it today, half of it tomorrow. In fact, you've got up to 4 weeks to fill the SpectroCHIP, right? And you can choose which SpectroCHIP suits your needs for that day. Our 384 instrument will run the 96 and the 24. The 96 will run 96 and 24. 24 will run just 24. Okay, germline genotyping is pretty easy. Let's move on to somatic mutation analysis and the challenges in oncology. So in oncology, we have a variety of desires or needs or questions that we wish to answer. You probably know what they are, but I'll tell you, all of them require a sensitive and accurate detection technology. Now, we've been working in the oncology space since back in 2004. Memorial Sloan Kettering were one of the first pioneers to introduce the MassARRAY platform for mutational testing for EGFR, KRAS, mutations in NSCLC and further expanded it to EGFR, KRAS and BRAF for colon cancer screening. They're a New York State approved CAP-clear laboratory. And what was our advantage over traditional Sanger sequencing? Well, traditional Sanger sequencing can only detect 10 to 20% mutation frequency, whereas we, in our standard biochemistry, go down to at least 5% mutation frequency detection, and Charles is going to show you a biochemistry that gets us down to 0.1% in a multiplex format. So what does a somatic mutation look like in the mass spectrometer? So this is from our collar Carter panel. The collar Carter panel was originally developed by Fumagalli and published in 2010. And of course, it's on our product list. You can pre-order this panel. And here we have a PIK3CA mutation assay. So we're looking at the red lines. The grey here is another assay in the multiplex. Right, so the red line, first red line is our primer that gets extended. If it doesn't get extended, you'll get a peak here. All right, but you can see this is now all being consumed into the single base extension products. So we're looking at the primer plus the mass of a G, T, and A. Right, so what we've got here, our A is our wild type for this particular nucleotide, 95.2% frequency by measuring the area under the curve. Here, the G is one of the mutations, and it's the one that we're observing in this sample, and we've got a 4.8% PIK3CA H1047R mutation. So you can see how clear that is from the baseline of the mass spectrum. So that's our collar Carter panel. Our collar Carter panel has 6 genes, 32 mutations in those genes, has been clinically validated in a variety of groups, using only 60 nanograms of DNA per sample, detecting mutations down to 5%. And of course, short amplicon works on FFPE. Another panel that we've been working on is our OncoFocus panel. So our OncoFocus panel is for detection of mutations in KRAS, BRAF, EGFR, NRAS, and KIT. In the United States and Australia, we call them the reimbursable genes because they have approved targeted therapies for which they're companion diagnostics for. Right? So we have a decision, a therapeutic decision that is clearly made based on the mutational profile in each of these genes. And those, the specific cancers that have therapies approved are lung, colon, and melanoma. We've optimized this into a 2-well global PCR approach such that it only requires 20 nanograms of DNA. And I just told you a lie. It only requires 2 nanograms of DNA. But I know your DNA is degraded. So I'm going to tell you 20 nanograms because I want a buffer. But how degraded is your DNA? What's a nanogram? A nanogram could be nothing. A nanogram could be a whole bunch of nucleotides. Or a nanogram could be nice, high, kilobase size, high molecular weight DNA. And this is a challenge that we run into. We're highly robust. Yes, sir. Both point mutations in insertions and deletions, and that's on the next slide. So, yeah, you'll get a detailed overview of exactly what mutations it covers. I agree with you. Saying just the genes you cover is ridiculous. Being reimbursed because you do 1 mutation in a gene at the same level as you get reimbursed for when you do hundreds of mutations in a gene is also absurd. So I know the sensitive point, and I will certainly demonstrate it. All right. Now, the landscape changed early this year. In colon cancer, CAP, AMP, ASCO, and ASCP, based on what is compelling evidence, released draft guidance to change the screening, the companion diagnostic guidelines for cetuximab therapy. And the draft guidance was, instead of just looking at KRAS exon 2, we should be looking at KRAS, NRAS, exon 2, 3, 4, and these other specific codons, as well as BRAF V600 mutations. Now, we did have most of those in our OncoFocus panel, so just to show you how this panel has evolved and the spectrum of mutations that it has in it, we've got the 5 genes, we've got more than 300 mutations, both SNPs and insertions and deletions. In the black is our version 1 panel. which we released based on the NCCI guideline content in the 1st of 2014. There was then an extension added in the 1st of 2015 with some additional rare BRAF mutations as well as KIT mutations which had been included in guidelines for melanoma. Then, of course, this year and just last month, we released the extended content based on the new guidelines that were released earlier this year. So you can see we are focused on the guidelines. We're focused on maintaining our panels and making sure that they're state-of-the-art. Now, you can see the content. These are the single nucleotide and multinucleotide polymorphisms. These are the insertions and deletions in exon 19 and exon 20, BRAF, KRAS, NRAS, and KIT mutations. You can see the depth of mutations in EGFR. And that's because EGFR is a nasty little beast. EGFR in the Caucasian population is reasonably simple. You can pretty much cover 90% by just looking at L858R and the 15 base pair deletion. It's in there somewhere. Too early in the morning for my eyes to see that. The 747 to 751 deletion. But in the Asian population, it's not the same. In the Asian population, we actually have an incredibly high rate of EGFR mutations, but also a broad spectrum of EGFR mutations, meaning that panels that were developed for Caucasians miss a significant number of actionable mutations in the Asian genome. Look at the difference, 10% frequency in Australia and America. Wow, Vietnam, 64%. India, nice and high, 26, 22. And the reason I say nice and high is because EGFR is a good thing to have in non-small cell lung cancer. It's sensitized to EGFR tyrosine kinase inhibitor, an inhibitor that is proven to increase progression-free survival and does have minimal side effects. China, 26, 22%, Chennai and Mumbai, and in fact we just heard about a study that will be published for Kolkata where they're up in the 30 to 40%. Right? So significantly increased here in India, but also across the rest of Asia, meaning that we really do need that broader distribution of mutations in EGFR for this region of the world. All right, so let's have a look at how we detect these indels, because indels are tricky. They're tricky on many technologies, tricky on next-gen sequencing, tricky on real-time PCR. So how does the MassARRAY detect them? So this is the region in exon 19 of EGFR that is commonly deleted, and many of these deletions are activating. All right, so they're gain of function. So we've got 16 amino acids, 48 nucleotides. We can have a deletion from here, our 2,236 coding base, through to 2,250. Activating mutations need to be in frame, so they're in multiples of 3. Right? Not any random mutation can cause an activating mutation. A frame shift is a silencing mutation. So they've got to be in multiples of 3. So here's an activating mutation deleting 5 amino acids, 5, 6, 7. Now, let's have a look at it. Single base extension chemistry. Anneal a primer in here. Wild type will be a G. First mutation is an A. Second mutation is a T. Third mutation is a C. Our primer, 5,000 Daltons plus G, C, A, T, all has different masses. So we can see all 4 potential products and therefore detect all 3 as well as the wild type in that one assay. But it's not that simple, right? There's not just 3 indels in this region. We can have a further deletion of 18 bases, a deletion of the same 18 bases and an insertion of 3, a deletion of 21 and an insertion of 3. So what we do is we do dual detection, dual confirming detection and discrimination of the insertions and deletions, such that for these 6 examples of mutations, we have 1, 2, 3, 4, 5 assays in multiplex format. So 6 mutations for 5 assays sounds like a bit of a gain to me. It's starting to grow some efficiency. One of the real-time PCR would require 7 assays. One for each mutant and one for the wild type. And they're difficult to multiplex. So that's why the real-time panels are focused just on the limited number of Caucasian mutations. Otherwise, you consume way too much DNA. Now, fragment analysis is another, method of analysing this region. Fragment analysis is you amplify the region with fluorescent primers, run it out on a capillary sequencer and see if there's a smaller product. Simple, right? Simply explained. The problem is, that's a 15 base pair deletion. So's that. That is absolutely no question sensitized to EGFR TKI. This one, we don't have any clinical data on. We make an assumption. Bit of a dangerous assumption, actually. To assume that a deletion of 5 amino acids and an insertion of 1 is an activating mutation when it's only been seen in a few lung cancer samples is a dangerous assumption. Now, it may work. It's worth putting them on the therapy, but you want to watch them very closely. And if you can't tell the difference between this and this, who do you know? How do you know who to watch? This one here is 18, this one here is 18, this one here is 18. The reason this one's 15 is it's 18 but a gain of 3. Loss of 18 and a gain of 3. This is loss of 21 and a gain of 3. Same story again. Known to be activating, assumed to be activating, no clinical data. But it's, again, not that simple. There's not just 6. There's more than 100 mutations in this particular area of EGFR. 100 net losses or deletions. And we characterise in dual direction those 100 mutations with 12 forward, 16 reverse assays. With that 2 well global PCR only requiring 20 nanograms of DNA into it, right? Looking at all 100 recognised deletions in exon 20. Just next door, he's equally as nasty. Generally net gains or insertions in exon 20. Many of these known to be resisting to the TKI therapy, and we characterise the 40 that are known in that area with 8 forward and 12 reverse. So that's how we get this diverse content in a multiplexed format. So if we have a look at the FDA-approved TheraScreen panels, the content's in red. You can see we've broadened the content because we know there's a lot more mutations that occur in Asia at a higher frequency. Now, I said we only need 2 nanograms, theoretically, if you have high molecular weight. I told you to put in 20 nanograms. 20 nanograms could be nothing in a worst case scenario. 20 nanograms could actually be 30 because generally OD is pretty close to a random number generator. So we want to know, because we don't have material to waste. We have limited amounts of sample. We want to know exactly how many intact molecules of DNA template are present. I don't want to know nanograms. I want to pick up a 5% mutation, I want to put 300 to 500 copies of DNA template into the reaction, such that I've got somewhere between 15 and 25 copies of the mutant in there, because we get this thing called sampling noise, right? When you take a sample of DNA, it is somewhere on a bell curve, right? If I put 20 copies of DNA in a reaction and I have a 5% mutation, how many copies of the mutant DNA did I put in the reaction? Come on, be brave. 20 copies, and it's 5%. So what's 5% of 20? 1? That's what the accountants will tell us. Yes? 1? A biologist will say, well, that depends. Because sampling noise means that 33% of the time, I actually sampled less than 1 mutant. And 66% of the time, I sampled more than 1 mutant. Well, again, the accountant will tell you that less than 1 mutant is 0.99. And a biologist will say, well, you can't have 0.99 of a molecule, so it's 0. Right? So we need to make sure that we're not down in this area of sampling noise. Right? Sampling noise is, India has 50% male, 50% female. This particular section, Actually, you're pretty good. You're right on target. Let's go with rows. The first 2 rows, 100% male. The next row, 100% female. Sampling noise, right? We have this random distribution. Well done. Representative. All right, so what we did is we built a panel because I had 2 needs in our service laboratory. Number one, when I get a DNA sample in, I have to trust the name on the barcode. And I've got it shipped from somebody else. So I had a need to protect myself and to ensure that when my customers challenged that I might have mixed up a sample, that I had a way to control for that. But I also needed to know the number of intact molecules. Right? DNA looks like DNA, right? Whether it's come from me or come from you. It's a liquid in a tube. So what we did is we built a panel that barcodes the DNA. 44 high minor allele frequency SNPs. Yes, they are high minor allele frequency here in the Indian population as well. High minor allele frequency SNPs gives us power of discrimination of 10 to the minus 18. You'll get put in jail at 10 to the minus 14. Right? Power of discrimination, that's the forensic panels. We built in extra power because we're trying to match tumour to germline. And we've got a thing called loss of heterozygosity, which effectively makes half of the markers uninformative because they're heterozygous in the germline and therefore could be anything in the tumour because of loss of heterozygosity. So you need significant power to do that. So 44 SNPs, 10 to the minus 18 power, 3 sex markers, tells you the sexes of the samples, and then we have these 5 assays in here that tell us 2 things, 2 very important things in the clinical sample. How many amplifiable molecules there are, and whether there's a PCR inhibitor present. Because we use a process called competitive PCR to quantify the absolute number of DNA template, which means that we put a known competitive standard in the reaction. If you get low yield against the competitive standard, then you've got a PCR inhibitor in place. All right? So it tells us those 2 things. Completely automated matching and database software. Builds a historical database. You see a sample today that you saw 3 years ago, it'll tell you that. Right? Because it maintains its database and it matches new samples to everything in its database. It says, okay, you saw this sample 3 years ago. 3 years ago it was called Rakesh, and now it's called Niraj. But it's the same DNA sample. So potentially you've had a mix-up. Or you had a sample from this hospital 3 years ago under this user ID, right, that was the same name, but they're now a different barcode. They're now a different genetic barcode, right? So it protects you from those matches and mismatches. If you're ever questioned, you can always go back to a patient, grab a buccal swab, match their tumour to their buccal swab, and confirm that this unusual mutation that's normally only seen in melanoma and now is seen in a lung cancer isn't a sample mix-up. So it's got that completely automated analysis, but for the clinical samples, it's the number of amplifiable copies. I want 300 to 500 copies to go into my reaction. How many copies have I got? Well, the MassARRAY technology only requires 100 base pair amplicons, right? So I'm looking at the copy number here because what it does is it tells you the copy number across a size ladder from 100 through to 500 base pairs, right? These are true, real FFPE samples. At 100 base pairs, I get just over 4,000 copies. Plenty of material. At 200, I've got just under 2,000. At 300, I've got 700. So maybe you want to do an exome sequence. And maybe, I don't know which technology you're using, which biochemistry you're using, so I'm just going to use some guesses as examples. But maybe you need 10 nanograms at 300 base pairs, right? 10 nanograms being 3,000 copies at 300 base pairs. Well, you haven't got enough. You're going to have to put in 30 nanograms to get the material that you need to generate your library, right? That's how it works. And this one down here, 250 copies at 100 base pairs. Oh, I said I really would like 300 to 500. 250 might be just enough, but probably add a little bit more into the MassARRAY technology. Don't bother running anything else. You just don't have it, right? You've got like 70 copies at 200 base pairs, despite the fact that via OD, we put in 10 nanograms of every one of these samples into this reaction. So now you can match your sample quality to your downstream analysis technology. Not wait until your sequencer fails, right? You say, oh, I haven't got enough for this. I'm going to have to run short amplicon to get any result for this patient. So let's focus on actionable genes, short amplicon, small amount of input material, because this patient doesn't have enough material for a big extended study.
So you could use this for cell-free DNA?
You can also use it for cell-free DNA, because with cell-free DNA, it's multiples of 166 base pairs, right? And the biggest concern in cell-free DNA is that you've had white blood cell lysis, right? And therefore, if you've had white blood cell lysis, you see a flat line. With cell-free DNA, you see a pretty rapid drop. You see your 100 base pair amplicons. You will see a few 200s, but they're pretty low, and nothing after that.
So that's exactly what. So if there is a white blood cell lysis, so
You have the high molecular weight material. And that means that you've contaminated your sample with normal cells. You're either going to have to go real deep, or in all reality, I believe that the sample is lost to science. Yeah. There's a lot of collections out there, right? Clinical research collections where they've collected plasma, but they didn't collect it with circulating cell-free DNA in mind. They collected it with sort of protein or serum, or, analysis in mind, popped it into the biobank. We don't know how they collect it, so we are able to QC those samples to see which ones should be included in a clinical study.
So just curious, so you have any data on your cell-free DNA sample?
I do have data. I don't have it in this particular presentation.
So for the low fetal fraction cases?
Sorry?
For any low fetal fraction cases?
No, I've got data on. Yeah, I don't have any data on NIPT. I have data on circulating cell-free DNA in normal samples. Yeah. Think of them as non-pregnant females, I guess, but females and males. All right, so we have this extended panel, which is now up to date with the newest guidelines. So the exon 3, exon 4 content in KRAS and NRAS for colon cancer. We teamed up with South Bend Medical Foundation, and we did a clinical validation study. So 259 colon cancer FFPE samples that had been previously run on alternative technologies. So we had orthogonally tested all of these samples, and what did we find? Using our extended OncoFocus panel, so the V3 OncoFocus panel, we found RAS mutations in 41% of cases. All right? Stop there just for a moment. What happens with the 60 and the 41%? Let's think about this. Well, the 60%, these are point of diagnosis samples, colon cancer samples. They don't have RAS or RAF mutations for 60% of them. Doesn't matter what else they've got. They get cetuximab, right? No question. There's no other question that we need to ask of these 60% of these samples. They get cetuximab. The sad news is they fail cetuximab eventually. Then we need to ask serious questions as to what we can do at that point. For the remaining 41%, then we need to ask detailed questions of what else we can do. Can we use another therapy? Can we use a therapy that's approved in another tissue, so on and so forth? Right? And this is the way that the MassARRAY and the sequences work together. Right? Because we're significantly lower cost than next generation sequencing. In colon cancer, we'll detect 60% of the patients that you know at a lower cost where they go. But don't get scared. I know you guys like next-gen sequencing. Don't get scared. They will come back, right? They will fail cetuximab therapy. The very sad news of it is they will fail cetuximab therapy, at which point they need a detailed analysis to work out if there's anything left to do with those patients, right? And of course, the 41%, of course, need a detailed analysis at baseline. Now, by extending the content, what we did is we picked up an extra 15% of mutations, right? So by having that extra exon 3 and exon 4 content in those two genes, we picked up an additional 15% compared to the KRAS signature FDA-approved test. 15% of patients were going on to a very expensive therapy when they actually could have been having some mints, right? Because they already had their resistance markers. Okay, lung adenocarcinoma. So lung adenocarcinoma has a very clear testing paradigm. KRAS testing, EGFR testing, bad news, EGFR TKI therapy, which is pretty good news. Next in the steps, so you can see that our OncoFocus panel, right? We've got KRAS, BRAF, NRAS for colon cancer, KRAS, BRAF, NRAS KIT for melanoma. We've got KRAS, EGFR for lung cancer. It's because a lot of our hospitals like to just have one test, single workflow for the three big cancers. So we've got that covered in our OncoFocus, but we also have that as well as some other mutations covered in our lung CARTA panel. But what I want to move on to is our ALK translocations. So ALK translocations along with RET and ROS1, right? ALK kinase inhibitor therapy, or the inhibitors for RET and ROS1, are included in our lung fusion panel. So with these fusion genes, the current standard is FISH and potentially IHC, but FISH is certainly the FDA-approved companion diagnostic for crizotinib. Now, probably don't need to tell you how FISH works, so let's keep it fairly simple. The way FISH works, red probe, green probe, start of the gene, end of the gene. If it's broken, they go apart. If it's not broken, they're together. Simple enough. So what we're looking for is where they're together. You see them together with a little bit of yellow in the middle, right? And where they're broken, you see them apart. I, like probably several people in this audience here, spent years looking down a microscope at coloured lights, right? And I know how difficult this is. The technician needs to go through, look at the cell. Does it look like a tumour cell? Yes, it does. Look at the probes. Are they broken? Are they not? Broken. Strike one. And then we do that 50 to 100 times. Right? Because you need to detect 15% of the tumour cells, those that look like tumour, having split signals before you can call it a positive. Right? So for 15%, you've got to do 50 or 100 cells. And it takes quite some time, at least an hour. Now, we were given the challenge because PCR techniques don't detect de novo fusions. The beauty of FISH is that it doesn't detect what it's broken with. It doesn't detect what's been put in front of ALK. It just says ALK's broken, right? Whereas PCR techniques, you need to know what it's broken with. And unfortunately, although ALK has a partner that it seems to like, EML4, it's highly promiscuous. It also likes KIF5B. It also likes CCDC6. It likes anyone who has a highly expressed promoter, right? So we were given the challenge to detect known or de novo fusions in ALK, RET, and ROS1 in a high-throughput, cost-effective manner using molecular biology, using PCR. And we achieved it, right? So we now have a panel that will do this using only 5 to 10 nanograms of cDNA. So we're using RNA that's converted to cDNA in a single reaction. RNA from FFPE can be a bit tricky, although I'll show you it's not that tricky, but we do have a variety of controls in there. And in fact, we have a single workflow process. Tissue, total nucleic acid extraction, pull out the DNA with a DNA column, DNase treat the flow through, convert the RNA to cDNA. So you actually come off the same curl. How does it work? Now, I showed you in sample ID, we can quantify numbers of molecules, right? We can say whether it's 100 molecules, 1,000 molecules, so on and so forth. And we do that using this competitive internal template. A long oligo that's the same sequence except for a single base as our target, right? We synthesize it, so therefore we know how many copies there are. So we design an assay to areas down near the promoter, exon 1, exon 2, exon 2, exon 3, right? And areas just after the kinase domain, right? Kinase domain in ALK is exon 20, right? So 20, 21, 22, 23. Then we quantify them, right? Now, if this is intact, if it's one entire transcript, then the numbers will all be the same. And they should be low most of the time, because this is part of the cell proliferation pathway. But if they're exactly the same numbers, maybe it's 100 copies, 100 copies, 100 copies, 100 copies, ALK's intact. But what happens when ALK's broken is that this area is under the control of the different promoter, right? So we see low copy number of this and high copy number of that. So what does it look like? This particular peak here is from our standard, 250 copies. This particular peak here is from there. There's nothing there, right? So we've got no transcript down here, and the opposite is up here. That's from the standard. That is from the transcript, right? Keep it simple. Nothing, everything, right? Few molecules, lots of molecules. You can't be intact, right? And importantly, the kinase domain is being expressed. So we have this setup in all 3 genes for lung adenocarcinoma that we call our lung fusion panel, ALK, RET, and ROS1. ALK is an approved therapy. RET and ROS1 are tracking probably the fastest of any drug down a clinical trial. So they're not far away. And we teamed up with the Peter MacCallum in Melbourne to do a clinical study. 51 lung FFPE samples and 6 control lines were supplied by our collaborator. We did a comparison between 5 molecular methods, right? Lung fusion, nanostring, Archer, Nugen, Oncomon, along with FISH and IHC. I'm not going to show you all the data because that would spoil the paper that we're preparing. I'll show you a little bit of it, right? So here's the pile-up or the Venn diagrams for FISH, lung fusion, and nanostring, right? So let's start with ALK. Now you can see these are highly enriched. Our collaborator sent us samples that they were retrospective samples that they detected high numbers of ALK, RET, and ROS1 translocations, as well as a few wild types in there as well. Start in the middle. The middle's easy. 20 samples detected by both molecular techniques as well as FISH, right? And then we go to the outliers. Let's start down here. 1 sample was detected positive as FISH and not seen on either molecular method. Now, look at the hashtag. IHC negative. So the FISH was positive and the IHC was negative. Have we got a FISH false positive? Or have we got a broken ALK, but where ALK doesn't have an express promoter in front of it? It's possible. There's a lot of passenger mutations that go on in cancer. Not all of them are driving, right? Not everything that occurs in a cancer cell is driving. And maybe it's got a passenger mutation that means the FISH is positive and the IHC is negative because there's no protein produced, but the gene could be broken. I don't know the answer to that. We haven't elucidated that yet. But I think it's pretty clear that without a protein, it's not driving. This one here, 1 positive sample detected by both molecular methods, not detected by the gold standard. Ooh, what are we going to do about this? When we went back, well, first of all, ran the sample on IHC. IHC is positive. I know you'd like that. And also, when they went back and had a look at the slides for the FISH, or at least the camera images for the FISH, there was a narrow split. A really narrow split. Right? So that certainly looks like we've explained all of this. And in fact, it looks like we've explained that the 2 molecular techniques are better than the gold standard and than the gold standard of FISH. All right, moving on to RET. Nothing in the middle there. 1 sample positive on FISH and lung fusion. Not detected on nanostring. It didn't have a large number of intact molecules, and therefore it was detected on our variant panel. Is the caveat there, but it was missed by the nanostring. Nanostring's got 10 false positives, so we've got a significant problem with nanostring and FISH going on there. Third, ROS1. 3 samples in the middle, 2 positive on FISH, not positive on the molecular techniques, coming down to the double star. Going back to the images, it wasn't actually a split, it was a loss. It was an atypical 3' loss, which means either the 3' probe didn't hybridize or the 3' probe region has been deleted, right? But it's not actually a break apart, right? So I think we've explained all of that.